Yeah, so now we are on. Hello, everyone, and welcome to BioElevate for 2026 for this year. And thank you very much for joining us. And as you know, maybe this is a series for highlighting exciting work from early career researchers in bioelectronics. And we are trying to create a space for global scientific exchange. And as you know, it's just a free and online, and we are happy to have people from around the world. So just a reminder about when BioElevate is happening. So for this year, BioElevate is happening every 3 weeks on Tuesday, but we have changed the time for this year to be 4 p.m. instead of 2 p.m. UK time. And if you haven't already, we will share at the end of the talk a mailing list so you can join the mailing list and then you can get an invite about like what are the next speakers and who they are. And we are going to announce our next speaker as well by the end of the talk of today. So please stay around for that. And now I'm very happy to and excited to kick off the BioElevate series for this year with our first speaker, Dr. Tyler Cannon from MIT. Dr. Tyler, she is a postdoctoral fellow in MIT Bioelectronics Group and incoming assistant professor of biomedical engineering at the University of Minnesota. Her research focused on developing endoscopic optical imaging system to capture neural signaling in the central and peripheral nervous system and functional alterations in the context of diseases. So she's going to give us a talk today on a fiber-based optical imaging platform for functional central and peripheral neuroimaging in vivo. So we will jump into the talk directly, but just a quick note. If you have any questions, please write them in the Q&A box or raise your hand at the end of the talk, and we are going to take all of your questions at the end of the talk when it's finished. So now I will jump into Tyler. Tyler, please, the floor is yours.
Great. Thank you so much for the introduction. I will go ahead and start sharing my screen. Great.
Yeah, we can see it.
Presentation. Awesome. Yes. So great. So thanks so much for the invitation to participate in this series. It sounds really exciting, and I'm looking forward to seeing what you have coming up in the future and participating. But as Latifa said today, I'm excited to present some of our recent work in the MIT Bioelectronics Group using fiber-based methods to perform optical imaging of neural activity in both the central and peripheral nervous systems. And so I wanted to just start off by highlighting some of the advantages of using optical imaging as a neuromonitoring tool, which include being able to capture very detailed dynamics, including single-cell resolution of specific neuronal populations of interest at very high spatiotemporal resolution, thanks to the very bright and fast fluorescent indicators, such as GCAMP, that have been developed to be able to provide an optical readout of neuronal activity. So you can see those cells flickering there. And also thanks to endoscopic probes that have been developed to be able to be inserted and implanted into the brain so that we can view these neural populations not just in superficial areas, but also in deep brain regions where light otherwise wouldn't be able to access. But that being said, there are a number of drawbacks to using endoscopic probes, including the very restricted fields of view that are possible to monitor these cell populations over. And so this is usually restricted to a single brain region, which is somewhat at odds with how we would ideally want to be able to monitor the interaction of different populations of different regions of the brain along a circuit. And then this challenge becomes even a bit more fraught if we're moving into also parts of the peripheral nervous system, including the enteric nervous system, which innervates the gastrointestinal tract, which spans these really large areas in this very delicate and dynamic tissue environment that's pretty incompatible with most of our existing optical imaging tools. So our overarching goal with this work is to begin to overcome these challenges and to be able to extend the very detailed high spatiotemporal resolution that optical imaging can provide to be able to more broadly monitor these brain-body environments. So as I mentioned in my title slide, fibers, for the past several years, the MIT Bioelectronics Group under Polina Anakieva has been leading the development of soft polymer-based fiber implants for neural interfacing, incorporating not just one, but typically multiple modalities for both stimulation and recording of neural activity. And so we make these probes using thermal fiber drawing, a very scalable process which begins with this macro scale preform. So note the big scale bars here, which we fabricate just using bulk machining procedures very cheaply, and then apply heat and force in our thermal drawing tower to be able to dramatically, controllably reduce the cross-section of these fiber devices and producing these hundreds of meters of functional fiber that now have features on the micro scale. And by incorporating different materials, we can produce many different fiber geometries with some examples here. Each of these materials enabling different functionalities, which I'll illustrate with one of our recent fibers, which we termed the polyfiber for the polymer optical, electrical, fluidic, neurological interface. And so we, again, were able to incorporate a number of diverse materials to enable electrical and optical functionalization, as well as fluid delivery into the brain. So in total, we're using just a single soft fiber that we're able to produce at large scale and be able to perform drug and virus delivery, electrical and optogenetic simulation, and electrophysiology, as well as fiber photometry, thanks to all of our back-end interfacing. And I'll highlight photometry here as well, since this was the first time we were able to do this with these polymer-based optical waveguides that previously had been too lossy to be able to both transmit but also detect light in return, where, again, our signal of interest is these populations of neurons that are expressing fluorescent indicators such as GCAMP. And so we send our light in, and then we're able to get this bulk readout of our optical analog of neural activity. So we were able to demonstrate all of these different functions all at once in mice, where we were implanting a fiber into the nucleus accumbens, which is part of the mesolimbic reward pathway, a major site of dopamine release. And so we were interested in being able to study that pathway in a bit more detail. So we expressed both a channel rhodopsin for optogenetic stimulation of the accumbens, and then also a dopamine indicator GrabDA for detecting this neurotransmitter. And so we found that, again, with just a single device, all in the context of a single recording, we were able to optogenetically drive our neurons of interest. We were able to get this photometric signal for this fairly slow release of dopamine in response to typically electrical stimulation, as well as detect our fast spiking behavior. So getting this kind of nice picture of both faster, slower dynamics of cellular specificity and more broadly in this neuronal population. But to consider our fiber photometry capabilities a little bit more closely again, so it's a very straightforward way to access the bulk behavior of this neuronal population, or as I just showed, through neurotransmitter release. But compared to actually performing spatially resolved endoscopic imaging via a probe, typically an endoscopic or graded refractive index lens. With fiber photometry, you're effectively just averaging out signal over a relatively large area, getting a single trace, versus if you're able to actually detect the behavior of individual cells, you can build up these signals and get a lot more power from these observations quickly. But on the flip side, the graded refractive index lenses are usually a bit larger, but more problematically much stiffer than our soft polymers. And so we see a much greater immune response, both acutely and then chronically, to the stiffer materials in the brain, motivating how we would potentially be able to stick with our soft polymer-based devices, but be able to incorporate spatially resolved imaging into these. And so that is exactly what we are trying to do with our approach being using polymer-based optical fiber bundles as flexible neuroimaging tools. And so these work by, I'll go into more detail, but using thousands of individual waveguide cores instead of just a single optical core to be able to independently transmit our spatially resolved signals. And so I'll show some of our results in both the brain and in the enteric nervous system and get started with brain. And so, yeah, I'd like to just explain how our fiber bundles work a little bit more explicitly compared to showing here just our step index fiber photometry fibers, which incorporate just one optical waveguide, which even over short distances, if you have a spatially discrete signal at one end, that light gets scrambled as it travels through the multimode fiber to the other, resulting in just this blurred fluorescence output versus if you have a fiber bundle. Each of these individually arrayed waveguide cores is able to independently transmit that fluorescence information and then give us this somewhat pixelated but still discretely resolved fluorescence output. So here's an example of what some of these devices look like, where an important parameters to note would be that your field of view is going to be dependent on the diameter of the fiber bundle. The resolution is going to be dependent on the individual size of these fiber cores. And here's just an example of looking at a fluorescent target, being able to get, again, kind of the macro scale view of our sample structure, and then zooming in to see the light that's coming through each of the individual fiber cores. And so we've been working on fabricating these bundles ourselves, and I alluded to having thousands of individual tiny fibers, which is a little challenging. So I'll touch on the procedure that we've been developing for accomplishing this at scale, which initially sees the thermal consolidation of say a reasonable number on the order of 60 polymer waveguides that we thermally consolidate so that they stick together in a cohesive, well-ordered bundle, run that through our thermal drawing tower, and then you get these about a reduction of 10 in terms of the size of these devices. But then we can cut up this first step fiber, rearray them into a secondary matrix where now we're on the order of 30 by 60 or 60 by 60, so getting into that thousands range. Run them through the draw tower a second time, and then start to achieve these numbers of cores that we're interested in to be able to effectively monitor neuronal activity. And so we can also, incorporate our multifunctional elements. So this being an example here of inserting some channels through which we can either use as fluidics, or we can also incorporate, as I'll show in the next slide, microscale electrodes for electrical sensing and simulation. That being, again, the case in this 3000 core fiber that we were able to draw. Even despite our integration of electrical components, we were able to image fluorescent samples through the tip, again, just by focusing on the opposite end of the fiber that's interfacing with the fluorescent sample. We can interpolate between these discrete cores to be able to get a smoother signal output with less of that pixelation, as well as etch off some of our protective coating to be able to expose, in this case, just a couple of electrodes out of the coating, and then perform our electrical and optical connectorization so that we can interface with all these different elements. But that being said, the rest of the results that I will show for the remainder of this talk have just been taken using commercially available polymer optical fiber. So while we work on getting our more multifunctional devices online and ready for in vivo use. And so we've been exploring the usage of our fiber bundles as, of course, neural implants in our mice, where we can use them sort of analogously to either other implantable neural devices or optical endoscopes. And so here I'm just showing what it looks like to see. Okay, my video is not showing up, but we're able to basically remove this magnetic dust cap on one end of the fiber and then be able to instead replace it with a small custom designed miniaturized microscope that the mice are able to wear on their heads. And then another aspect that we're really excited about with these devices is that we're able to co-house our animals due to kind of the robust and relatively small nature of these implants, which we like because we're interested in looking a lot of social paradigms where we don't want to stress our mice out by socially isolating them, which we have to perform with a lot of our other devices to preserve the integrity of those devices. And so one project where that's particularly important is this project that our grad student Pema has been leading. And this is looking at kind of the interconnection of stress and reward, but particularly in the context of drug addiction. And we know that it involves these multiple different pathways, with the paraventricular nucleus of the hypothalamus being more canonically associated with stress. And then at different sites, again, as I mentioned previously, our mesolimbic reward pathway, the ventral tegmental area to the nucleus accumbens, that the PVN projects to the VTA, but it's unclear of how these different brain regions might interact to respond to different stressful or rewarding stimuli. One of the reasons why this is challenging to study is because these are fairly spatially disparate regions. And so if you wanted to be able to look at them all at the same time with a singular probe, you would be taking out a very substantial amount of the mouse brain, especially because these are all quite deep brain regions as well. So we thought we would be able to leverage another aspect of our soft, flexible devices of not just having a better shot at preserving that local tissue environment with less perturbation, but also to be able to draw them and then to converge them together from multiple sites up to a single interface that we're able to use to be able to visualize all of our different brain regions at once. Where in these experiments, I'll reiterate this again, we're looking at a calcium indicator in the PVN, our stress region. We're looking at the projections from the PVN to the VTA within the VTA, and then we're also looking at dopamine release via D-light in the nucleus accumbens via 2 bundles and then just 1 step index fiber to see the neurotransmitter release in the accumbens. So what this looks like in terms of the actual device design is we design a box with a high resolution 3D printer that has 3 holes that are co-aligned to the coordinates of interest for all of our brain regions. So even though this is the particular question and application that we've chosen of interest here, you could actually customize it to be able to look at kind of arbitrarily any 3 brain regions, probably, as long as you're able to get a box that spans those regions. But in this case, these are pretty lightweight, small devices, where in this shot you can see, again, these 3 fibers that we then thread through our holes in our box and our custom ferrule so that they're all pretty closely co-located. And here's an example of what we're able to visualize here. Actually, that scale bar should be 0.2 millimeters, not 0.1, but some representative images looking into in our kind of blurred signal from our accumbens with the neurotransmitter, and then looking at our neurons in the PVN and their projections in the VTA, which I can show in a bit more detail here, as well as our mouse, fairly happy looking mouse despite having its 3 implanted fibers. We do perform these imaging experiments under anesthesia just because it's a bit easier for us to be able to focus nicely onto our fiber course, but in theory they could also be coupled with a custom microscope on the other end for freely behaving reportings as well. But here are a couple of videos of seeing those neurons fluctuate in the PVN, recording at 15 frames per second in this case. And also, again, those projections, a weaker signal that we pick up in the VTA, but that we are able to, we think, see some consistent structures if we do enough averaging over time. And using this in the general experimental context of interest, I will show our response in the accumbens to both saline injection, not too much, followed by fentanyl injection. These are both performed subcutaneously, so it takes a while before we're able to see a more broad response in the brain to these stimuli. But after a few minutes, we start to see the drugs kick in. And that similarly, we see more of a response in the PVN. It's a very twitchy brain region. Again, this is even under anesthesia. but to not only both of the injections, but then this more profound interaction after we start to see our kind of reward-related response to the fentanyl injection, as well as a somewhat coordinated, though slightly more unclear response from the projections that we see to the VTA. And so we're very much looking forward to being able to kind of further understand the dynamics between these signals and their interrelationship in this complicated circuit. And one of the ways that we've been trying to do that is by more explicitly looking at the signal in the VTA as well, rather than just looking at the projections from the PVN, while still wanting to get that information. And so what we did was we introduced, instead of just our calcium indicator into the PVN, we also introduced a red calcium indicator to the VTA, JRG Echo, and we're able to develop a back-end two-color, this time, imaging system. So we're spatially multiplexing both of these signals in our red and green channels onto the same camera, and then able to interrogate these different optical channels at the same basically frame rate that we were able to access with our single color recordings. And so what we were trying to do here was, again, all in the context of a singular recording, this is just showing from one mouse, was to see a coordinated response to similar and different stimuli. And so we didn't see too much of amount of activity at baseline in all of our various brain regions. When we introduced a predator olfactory based fear stimulus, we were able to see quite a bit of activation in the PVN as expected in our more stressy region. But following, again, in the same recording, the injection of cocaine to this animal, we saw, as expected, more activity in the VTA as well as the NAC along the reward pathway. But when we reintroduced the fear stimulus, we saw kind of the shutdown of some of this more reward-based signaling, but as well as a different activation. So all of these rows are sorted by spatial information in the same order between the different panels, where we no longer see the same response exactly to the fear stimulus as we did to the first time in the PVN before we also introduced a reward. So we think there's a lot of interesting information here that we're analyzing some of our data from additional animals and starting to try to draw some conclusions about the interrelationships between these brain regions. And then I'd also note that we were able to perform these experiments over fairly long periods of time. So we were able to kind of continuously collect data for more than 6 months after we were initially implanting the devices, with the animals tolerating these multiple implants quite well. So I'll wrap up the brain portion of the talk before I go into the enteric nervous system content. And yeah, we're excited about being able to kind of flexibly perform these multi-site spatially resolved recordings, and particularly for how well tolerated they were for longitudinal, potentially months-long different imaging studies that can allow us to really expand into more chronic monitoring that hasn't previously been possible with these tools. And with the time that I have left, I'll get into our imaging in the enteric nervous system, which I'm also quite excited about. And basically, we think that there's a lot of structural and functional diversity of the neurons in the gut, both in terms of their spatial heterogeneity and also their functional heterogeneity. And basically, they deserve the same level of sophistication of tools that we are using to try and understand neural activity in the brain. But again, the goal being, how can we actually develop our tools to be able to interface with this now very different environment? And so here, our platform is to be able to systemically deliver a virus to be able to induce fluorescence in even our peripheral nervous systems, including the enteric nervous system, and then interface, in this case, with the colon via an endoscopic bundle probe. So relatively similar to the brain work that I just showed more fundamentally, where we are inserting, in this case, non-surgically, just repeatedly and relatively non-invasively this probe into the mouse colon and then interfacing with it at the other end with our back-end multicolor endoscopic imaging microscope. And that this work was also led by our grad student Gary in the group, where we were using a retroorbital injection to be able to induce our viral expression of the fluorescent compounds in the enteric nervous system. And here's a more photograph-oriented representation of our fiber bundle, which we couple to both a rotary junction, so we're able to spin it once it's in the GI tract and capture a circumferential view and pulling that back of our gut wall, as well as a microfluidic channel along here that we are able to interface with the syringe port so that we can introduce various activity agonists. And so we start to see signal in vivo about 2 weeks after we perform our retroorbital injections as these fluorophores start to be expressed in the colon wall. This expression continues to increase, we see, for up until about 3 or 4 weeks, when then we think that at that point, probably the fluorescent proteins are starting to get trafficked out of the cell, and then we start to see a bit of a reduction, but still gives us kind of a comfortable month-long timescale to be able to perform these experiments in our expressing animals, which we can then validate that expression, also ex vivo collecting tissue samples, and confirm that we see these expression coming online after about 2 weeks, peaking around 3 to 4 weeks, and then beginning to reduce around 5 weeks. And so here's a more video-based format of looking at, in this case, our neurons that are expressing mCherry, which we were happy to be able to use an indicator in the red channel just to be able to avoid a little bit more of the background autofluorescence from the tissue. But we can also do imaging in green channel. So here's an example of where in real time retroorbitally injecting dextrin, which labels vasculature. In this case, what we're seeing is microcapillary beds in the colon wall moving around. And then thanks to our two-color imaging setup, we can also capture these two different signals at the same time to see both our neurons and our vasculature. But all of those were slightly more structurally oriented signals, whereas what we would really love to be able to do would be to do functional imaging in the enteric nervous system. So we've been working on using AAVs to deliver GCAMP to the colon in our mice. And so I'll show some data. As you can appreciate, maybe from the previous slide, we do pick up a lot of movement and motility in the gut. So this has also prompted a lot of motion correction strategies on our end, which here we're performing over a baseline recording. You can just see kind of a gentle wiggling of the tissue, but we're able to eliminate a large amount of that motility, at least towards the center of our imaging frame. And then following the introduction of a serotonergic agonist, just a laxative basically, and following our motion correction, that we can see a more profound change in fluorescence in these neurons that we were able to image here, still within a fairly stable field of view. And so by looking at this in a heat map orientation, and we can also see here what the data looks like if we don't perform motion correction and then average over 30 seconds or 300 frames, that here, again, we see a small amount of activity during our baseline, just some probable, normal endogenous motility. And then following the introduction of our serotonergic agonist, we think we're looking at various contractions here in the gut wall. We can average these and compare them to, getting a lot more information by being able to spatially segment out some of these putative cellular ROIs. I should mention that in this case, in this probe, we are able to see down to a resolution of about 10 microns. So we're not quite necessarily at individual neuronal imaging, but we think it's more than enough to be able to see the different features of the myenteric and submucosal plexus in the gut wall, as well as potentially some propagation of these waves across our field of view. So in these rows in the heat maps are organized spatially, and we do see a small amount of propagation as we go along with each contraction. So we're really excited about being able to use this platform as well, to be able to look at more molecular level orchestration of enteric nervous system dynamics, and particularly in the context of different both GI disorders and then also disorders where we have both central nervous system and GI comorbidities. And all of this work falls under our goal of being able to kind of expand the library of tools that we have for brain-body research. And with that, I will go ahead and wrap up and acknowledge everyone in the bioelectronics group, particularly everyone who is highlighted on this slide that contributed directly to the work, various funding sources, and as a personal plug, I will be starting my lab at the University of Minnesota this fall, so would definitely love to get in touch if you found any of this interesting. And thanks for the opportunity to present today.
Thank you very much. Thank you, Theo. It was a really nice talk. And yeah, it's super interesting. So now if anyone has a question, please write them in the Q&A box or raise your hand, and then I can allow you to speak if you would like to. So maybe meanwhile they're thinking about some questions, I can have some. Yeah, so first of all, I would like to ask you about the backend side of the fibers. How do you think, uh, easy or hard, especially if you want to do like free moving animal experiments? And yeah, and how did you do the assembly with the fibers? And can you comment on this, please?
Yeah, it's a bit challenging for sure, which is why we've been mostly sticking with animals that are under anesthesia for our current
Yeah.
recordings. But yeah, we had started by designing basically these customized mini scopes to be able to interface just with a single fiber, and so we were able to do that using these magnetically actuated platforms, and those are these battery operated devices that are writing to a micro SD card, so they're fully untethered, which was exciting. And so for especially for the work where we're using the kind of triple fiber probe, that it would, we think, just require kind of a redesign of what the base plate would look like, basically of how we would attach the microscope
Yeah.
onto the animal. And it would probably be a slightly taller device, which was kind of our only hesitation about being able to use those so far. But from a fundamental level, it's, yeah, I think just rethinking the geometry of how the mini scope would interface with the mouse's head. So trying to reduce definitely any of the weight and size of there as much as possible, but um, not a fundamental limitation in terms of being able to visualize the actual fibers.
Yeah.
It is, yeah, challenging to be able to adjust the focus for sure. So in our previous freely behaving recordings, we were still initially anesthetizing the animals to be able to kind of adjust the mini scope in the first place before letting them walk around.
Yeah, that's cool. Okay, so I really find like being able to image is super interesting because like we usually, especially like in many areas, that when we do devices into the brain, we just go blind and we don't know what's happening there. So I like that they are showing like some kind of, we can do, you can do also some kind of fluorescence for drug delivery as well, which is, I found it very cool. Yeah. Like, how can you comment like on how these things could be also useful for other type of things, like rather than just drug delivery or electrode recording, like, yeah.
That's a good question. And I think that one aspect is, yeah, being able to look at multiple signals at once, especially with the two-color recordings, which maybe is also slightly more complicated in the freely behaving context of a bit more hardware, but more pertaining to some of the results that I showed in the gastrointestinal tract. I think the possibility of looking at both neuronal firing and either neurotransmitter release or looking at that in concert with vascular dynamics would be really interesting, and some of that being thanks to all of these exciting fluorescent indicators that are constantly coming out. So practically, I think every neurotransmitter has a fluorescent indicator at this point, and I think that that will be really interesting as this field expands to particularly see that in freely behaving animals, of giving us more insight of, okay, so we know that these neurons are firing under this particular stimulus, but then looking at not just that, but also what the neurotransmitter dynamics are in that region as well.
That's really cool. So maybe if there is no question, we can close up and I can just introduce the next speaker for the seminar. Thank you again, Tyler, for the very nice presentation and for answering my question as well. It was really nice. So just I'm sharing my slide now. Hopefully you can see it. Yeah. So the next speaker of BioElevate will be in the next 3 weeks. It would be from University of Cambridge, from Britannia, and she's going to give a talk on Verstalife biometric 3D electrochemical device for monitoring cell growth and epithelial barrier integrity. And yeah, so if you are interested, please, you have the QR code here. You can register for the seminar. And a final thing is that if you would like to receive our emails about the next seminar speaker, please join our mailing list, and you're going to receive an email before seminars are happening. And with that, I would close up the seminar of today. Thank you very much, Taylor, again for the very nice talk, and hope we'll see you soon. And yeah, thank you very much, everyone, as well, for attending.
Thanks
See
for the
you.
invitation. See you.
Thank you. Bye.